Welcome to what is now the first colloquium talk of the semester because of our ICE cancellation. Very excited to introduce you today to Dr. Liz Chrastil, who's visiting us today from warm, sunny, not icy Southern California. She is here, like I said, from Southern California, UC Irvine, where she is currently an associate professor, but I think probably not for much longer. I think she'll be a full professor before long. She, before that, received her bachelor's from WashU in St. Louis, a master's degree from SELFS, PhD from Brown University, and then a postdoc stint with Chantal Stern at Boston University. A short stint at Santa Barbara, and then became a professor at UC Irvine, where she is in neurology and behavior, and also in the Center for Neurobiology of Learning and Memory. She's very well funded by both NIH and NSF to study individual differences in spatial navigation abilities. And what I like most about her work is her focus on individual differences. She studies spatial navigation and spatial cognition in kind of two major ways. So she studies structural and functional MRI studies of spatial navigation and path integration. But then she also has a separate line of research where she studies in kind of large scale navigation studies in fully immersive VR situations with complex navigational through navigational environments using MR. So it should be a very, very interesting talk today with individual differences of those things that I just said. So please join me in welcoming Dr. Chrastil.
Thank you so much. It's great to be here. Hopefully the Zoom audience or the Teams audience can hear me. Maybe say something. Can't. Oh no. What did I do? There we go. There we go. I think we're okay. Yeah. Okay, now I've got this thing on. Like at least all right. Hopefully we got the weird stuff out before things got too crazy. Okay. All right. So thanks for having me here. It's been delightful, wonderful talking with everyone and just hearing about what all your work you're doing and hopefully making some connections with some of the stuff that I'm going to show you today. And so we're going to start out. And my slides are doing something funny. Okay, so when we talk about learning a new city, we often walk around town, maybe wander a little bit, maybe we go from place to place. But so how do we learn the connections of streets? How do we learn, where we're going? It might be very different if you're moving to the city and need to know where things are for future reference when you're living there versus when you're just sort of like, there as a tourist. And also when you're just there, when you have to return to say like your hotel or Airbnb at the end of the night, you probably don't just like retrace your steps from all the things you did and go back, right? You've learned something about this map. We will sort of ignore GPS systems for the moment, but talk about, how you might find some ways to get around in shortcuts. So again, we're just trying to figure out, how do you learn these new places? And it's really important for a lot of reasons. And so a lot of the questions my lab studies is how does the brain learn spatial information? So how do individuals differ in those navigation abilities? I think we all know some folks, you don't have to identify yourself, but you know who you are. You can't find anything, you use your GPS at home every night, versus some people who know exactly where they are at all times, right? So there's this really big differences. And so we use, as just mentioned, a synthesis of cognitive and neuroscience approaches. So we use immersive virtual reality. So we have basically a big empty room where people walk around with a VR headset. We've also done a little bit of GPS tracking in the real world, and then neuroimaging. We have to use MRI and EEG, which requires a slightly different approach in terms of the behavior. But overall, again, we sort of try to combine these in different ways. But of course, our methods and what we do depends on the questions we ask. And so I'm going to sort of highlight what we're going to talk about here today. So first, just want to introduce you to why navigation is important to the study of aging and Alzheimer's. I know you guys are aging, a lot of aging folks here, so I wanted to make sure that that connects. I struck through this because we don't have time to talk, so I have a few strike throughs, but if you have questions, we sometimes talk about structure of spatial and non-spatial information in terms of the cognitive map. I'm not going to talk about that today, but happy to answer questions about it. How do we learn new environments? I'm also actually not going to really talk about that. But I will talk about how individuals differ in their navigation abilities, some of the work we've been doing on that in a couple different fronts. And then some questions about how the brain tracks spatial distance and how that relates to other types of relational distance processing and being a little more abstract about it. Not going to talk about how the brain tracks self-emotion information, but it's in there. I am going to talk about how our navigation abilities change as we age. And then sort of like a fun one, if we have time, is how does the brain change during some other life transitions, such as pregnancy? Hopefully we'll get to that. Okay, so first let's talk about, we'll do a little crash course on navigation and why it's important to the study of aging and Alzheimer's. So first, just to kind of introduce you guys to space and how we talk about navigation and some of the relationships of sort of memory. We have different types of spatial knowledge. And so one type, sort of basic type of spatial knowledge is called landmark, just salient objects that provide navigational cues. So the internet told me that these are some landmarks in Dallas. Hopefully these are right. Mobile statue, other things, right? So places you would know, you would say like, go here, turn left there, right? Things like that. The next sort of type of spatial knowledge we previously think about is route knowledge, which is just a series of place action associations. And so my favorite example is when I was in high school, my sister just got her driver's license. This was before GPS systems. We went to visit my grandma's house, which is like an hour and a half away. We didn't have a map or anything, but we knew once we got off the highway, we turn left at the stop sign, go to the vegetable stand, turn right, go to the school, turn right, cross the train tracks. My grandmother's house is on the left. So if there had been road construction or something else, we would have been in trouble because we wouldn't know of anything else about the area, but we just knew how to get there based on the turns. The third most commonly used type of spatial knowledge is called survey knowledge, which is map-like knowledge of distances and angles. So if we asked you to go from here, from the center to sort of main campus, UT Dallas, right, you could take a straight line shortcut and just go straight there through the highways, you know exactly which way to go and sort of direction. We can think of that as a coordinate system, like so it's everything is sort of the same no matter how you deal with it. But you might think to yourself, there's kind of a gap there between that route knowledge where you just sort of know turns and then knowing all this stuff. And so one area that we've been really interested in is something called graph knowledge, which is something in between that route knowledge and that survey knowledge, which is just thinking of things as a graph, which is a link network of place nodes linked by path edges. And so I'll put my points here. So this is, a graph of some locations. This is actually the same graph, but you can sort of stretch and shear it around. But it's because it has the same connectivity. The graphs are the same. And so you can think of this as like a subway map. You don't need to know all the twists and turns of a subway. You just need to know, take the red line 4 stops, change the green line, and go 2 stops. So we do a lot of work in this sort of level, again, which I'm not going to necessarily talk about today, but I'm happy to talk more about. So let me tend to make this connection with some of the work on aging and Alzheimer's disease, and why we should be interested in navigation, interested in talking about that sort of stuff. So first of all, there's a large overlap between the brain networks affected by early forms of Alzheimer's, and those are important for navigation. And so, in particular, the entorhinal cortex is really important for navigation. It's, of course, one of the earliest areas of Alzheimer's pathology. Hippocampus, retrosplenial cortex. These are all sort of some of the like most key areas for navigation, in addition to other things like perihippocampal cortex, visual systems, other things like that. So, but these are really some of the areas that we are sort of most important for navigation and also earliest affected by Alzheimer's. So that's one big overlap. And then we've seen that changes in navigation behavior and some of the functioning of entorhinal cortex can happen decades before the onset of disease symptoms. So a study by Kuntz et al. About 10 years ago now found that there were some behavioral differences, but also some genetic differences. So those who were ApoE E4 carriers had slightly different ways that their entorhinal cortex was functioning even at this young age, and that we could discover that based on these navigation tasks. And so using this navigation paradigm to look at entorhinal cortex function, we could see this change very early on. But essentially, we do see these differences between even young adults. And then those genetic risk, some groups have developed this mobile phone game called Sea Hero Quest, where you take a boat, you navigate around. And those folks have had, again, at younger ages, some slight differences in some of their navigation behavior for people who are at this risk. So in terms of looking at this map wayfinding, so basically they give you an overhead map of where you're supposed to go, and then you take your boat and you drive the boat along these different buoys. People with the E4 allele have longer distances wayfinding in certain levels of this game. They also, but they don't necessarily show differences in this other type of navigation. So interesting to see where, under what circumstances we see differences, again, at different ages. So again, navigation is early as its early marker. And another thing to note then is women have this twice of the risk of dementia than men, and we can sort of talk about it in different ways. It's not always clear exactly what that means, but one thing that is pretty notable is that there are sex differences in navigation that have been frequently observed, again, at young adults and throughout the lifespan. And so trying to understand whether this sex difference is also that important for in navigation, this might be important for this aging process. And we also know that sex hormones influence the organization of the campus. And so sort of putting that all together is one big question. So the aim of my group is really to characterize each individual's cognitive profile to understand, what's happening on the individual level, because sex is one factor, but there's a lot of other things, right? Your genetic risk might be one factor, other things, but there's a whole host of stuff going on. So we really understand people at a broad scale. So we're going to talk a lot about individual differences. Okay, so let's get into individual differences and a couple, sort of two approaches that we've taken with individual differences. And I'll talk about one in pretty good depth and another in just a little bit. So first I want to talk about a study that we've conducted. It's an fMRI study. We had about 100 healthy young adults participating in a navigation task. And so this is a navigation task where this is an overhead view of the environment. So there are some objects like tucked into these alcoves, and there's some paintings on the walls here. And this is what the participants see. So at each corner, they just see some arrows, and they just have to select, go straight left or right at each corner. So it's pretty straightforward to use the keyboard controls. And then we ask them, so they explore for about 15 minutes, and then we test them. And so we say, okay, start at the spaceship, go to the umbrella, and now they have to get around. But we've removed all the objects. So now they don't know that they've reached the right object. So people can and do get 100% wrong in this task. And they can also get 100% right. So anything in between. And so we see this large variability. And so people, again, are just navigating. And because they're doing this in the scanner, that's another reason we had a very simple task, it's not simple, but like a very simple controller, because we just wanted to make sure that they could do it and weren't focusing too much on like being able to use the controller, because they are going to be laying down and doing this task. And so in this case, they are trying to find the umbrella. This is, in fact, where the umbrella is, but they don't necessarily know that they got it right. And so they do this a bunch of tasks. And so what we see is a wide range of performance. So some people are down here basically getting nothing right. We call this chance. It's just one of 9, which is 9 objects. And then some people are doing really well at getting 100%, sort of everything in between. So we see this huge range, which is really nice for us. One of the things that we sort of decided a long time ago was rather than say like, how does the brain do this? There's so much individual differences that it was just, we couldn't really do that. So we just decided to kind of lean into the individual variability and sort of say, like, we're going to have some people who aren't going to do great, and they feel bad, and we say, no, that you're fine. We like to have that. So we do see that men perform better than women. And interestingly, there's a correlation with video game experience, even though, again, it's a simple controller. And we've done some work on this. We did a little bit of mediation analysis and actually found some aspects of video games are part of it, but it's not fully explained on its own. It does not fully explain the sex difference on its own. It's sort of via some ways of exploring. So yeah, and some correlations with some other types of some other sort of spatial abilities. But we wanted to dig into it a little more of this data, but there's lots of ways we can look at it. So first we wanted to look at just behavioral. So we wanted to say, like, how do people explore? And does that matter, right? So because we give people free exploration, that's another thing that is a little different from what we do. is that rather than giving people a set path to follow or that they see, right, people could go anywhere they wanted. And so it means that they, you know, might go to the same place multiple times. They might be more even in how they explore. And so we just want to see, first of all, on the behavioral side, does that matter, right? So does it matter how you explore? So one thing we looked at was this evenness. And so we called something called object exploration bias, which is basically just the standard deviation of object visits. So if you visit all the objects the same number of times, you would have like 0 standard deviation because there'd be no variation. But if you visit one object 10 times and one object 1 time, you'd have a big bias. And so we find that indeed, the more evenly you explore, so you have a lower object exploration bias, you tend to do better at this task. And we can look at it both in the objects and the hallways. And path entropy is a similar kind of idea in terms of evenness. And so we find this really happening. So this evenness of exploration seems to be important. We also found that finding the objects faster is correlated with accuracy. So the number of moves to find all the objects at one time is strongly related with performance. And in fact, by the time, if we group people by their performance quartile, by the time you get to like object 5, we start to see pretty big differences between, and I don't have the stars on this one, but we have pretty big differences between the top group, who's finding the 5th object here at about 25 moves, and the bottom group, which is finding it at more like 30 moves. And so basically, and it gets worse and worse as time goes on. So by the time they find their 9th object, there's some pretty substantial differences. So really, by here is where we can say about 5 objects in, which is about 3 minutes into your exploration, we can pretty much tell whether you're going to be a good navigator or like in the top half or the bottom half of the exploration. We put these into some principal component analyses, and again, found that these sort of components related to evenness are associated with this. Interestingly, we also then dug up some old data where we had active and passive people combined. And so, but it's not the case that you just give people a good exploration, they're going to be good at it, right? So it's not correlated. So the people who do well and people who are active, people who do well, who are passively guided by that same path, doesn't necessarily make them do better. So it really seems to be something internal about how you explore what you know, what you don't know that you know, how you're doing it for yourself seems to matter. And it doesn't matter just that you've given something that's really even. It's about you. We note that this also changes with age. And so looking at some of these younger adults with some midlife adults, we find that our young adults are not actually, our midlife adults are actually visiting objects more often than our young adults, but they are spending more time. So our midlife are spending less time in the hallways and spending more time at the objects, but they're not doing as well. So our wayfinding success in our midlife group is much lower than for our young adults. And then in this case, in our young adults, only the exploration quality measure came out to be related to performance in our young adults, but in our older adults, both quantity and quality were related. So there is something a little bit different. And again, these are early aging, so 45 to 55, people who, again, who probably played some video games in their lives, may have other things, but we're already starting to see some differences in terms of performance and in terms of how they're exploring and interacting with the environment. So then we can start looking at brain structure. So again, these people were doing this in an MRI scanner, so we can start to connect it with things. So we have hippocampal volumes. So we did correlations between overall volumes, but also hippocampal subfields for some folks. So we find that, there we go, just put them on. So we found that an overall relationship between hippocampal volume and especially anterior hippocampal cortex was related to performance. But I want to put a little asterisk in it here because it actually really matters whether you include, how you include sex as a variable here. So when you put the whole group in, even when we include it as a sex as a covariate, we find an overall relationship. But if you stratify by sex, they basically almost go in the opposite direction. And so we're still trying to figure this out. And even since in the year since this basically papers come out, we're really rethinking about how we deal with hippocampal volume and this like sort of sex differences, because sex is such a massive part of the relationship with performance. But trying to figure out in a better way. Yeah.
Just for information, are you controlling for total brain size?
Yes, exactly. So those are all in the covariates. Yeah, so include sex, age, and total intracranial volume. But still, we're seeing these interesting things, and so we're still trying to figure out exactly how to make this work. But we are seeing, some relationships. There's been a lot of controversy about hippocampal volume, whether it's related to navigation in sort of a healthy population. And so this suggests yes, but with like a really strong star. And we're really still trying to figure out how it might depend on a lot of factors. But we do see also some relationships with some of the subfields like dentate gyrus, which we were thinking about like this sort of a pattern separation task when you talk about this sort of complex maze environment. We also have other structures. So we also had white matter. So we had diffusion scan in this using diffusion spectrum imaging. And so we used a probabilistic tractography approach or a group connectivity approach to look at sort of a whole brain areas that are important that are related to, so where these microstructural changes are related to navigation. And here we found some of the areas we were expecting. So some of these hippocampal outputs. So hippocampal outputs in the cingulum bundle and the fornix were important for doing well at this task in these healthy young adults, as well as some other areas like the thalamic radiations and cerebellum were important for tasks. We didn't see as many sex differences here, although again, it was a vital piece of the pie, but they were a little bit more minimal here. But then we can also talk about functions. So again, they were doing this in the scanner, and we could talk about some network dynamics. And so we're working on this right now, thinking about how we can look how the brain changes over time during learning and during this task, right? Because they're experiencing this for the first time, and then they're being tested on their performance. And then again, some people are doing really well, and some people are not doing so well. So we want to see how these network changes across the course of learning happen on the overall versus as well as related to individual performance. So what we did here is we did a parcellation. So we used the Schaefer 200 and a subcortical map to parcellate the brain into sort of about 215 regions. And then we developed a coherence matrix with sliding windows. So we can do this, you can do a sort of the whole thing and just do like an overall coherence between brain regions overall, but we were doing these sliding windows of about maybe 2 minutes at a time. And so we can sort of go across and get a sense for how these are changing over time. And so we end up with this community assignments at each time point using this framework called dynamic community detection, which essentially says, these brain regions form one community, these brain regions form another community, and they might differ a little bit at each time point. So they're going to change a little bit as you go across time with some algorithms that help you sort of keep some things a little bit cohesive. And so we're interested in particular how things change over time. And so we're looking at this idea of flexibility, which will break apart into two different subunits. But flexibility is just essentially the number of times a node changes communities relative to the total number of changes. So if you see this middle one, this one starts in the red community, it goes to the yellow community, it goes back to the red community, and then it switches to this light green community. So it has several different switches over time, whereas this other one here in the corner stays in the light green community the whole time and is not being flexible. So it's been, others have found that flexibility is related to communication between communities, it's related to learning, and so learning rates are happening when some of these regions are more flexible. And then within flexibility, you can break that down a little bit. So we can break it down within whether or not brain regions are changing on their own or together. So if we again look at time point 1, we have say 4 different communities. And then this little node here switches from the yellow network to the red network at time point 2. We call that a disjoint change because it's moving on its own versus these 2 blue ones moving to the green one. We call that a cohesive change because they're moving together. So we can sort of break down flexibility within a couple of different regions because we're interested again in sort of how are these things happening and that might be informative. And so what we're finding when we go do whole brain disjointedness flexibility, we see that flexibility and cohesion are not particularly related to performance, but disjointedness is. So the higher disjointedness overall brain-wise throughout the whole thing is related to increased performance, right? And we're still trying to figure out why that might be or trying to understand that, but that's something that we're finding. And in particular, it's true in the exploration phase that disjointedness in the exploration is related to performance, but not necessarily as much during test phase. And we can break that down within the network level to find out, which of these canonical brain networks, so like default mode, control, vision, et cetera, are sort of driving this. And we find that in the learning during the exploration phase, we find that the visual network which is joining us, is related to proportion correct. Whereas during the test phase, the control network has the most relationship to performance. And again, there's also things that are happening on the group level as well. So we also see a lot of connectivity happening, but we were particularly related to individual performances as well. So in that sense, we've got a lot of individual differences with sort of one task that's a lot of people are doing. We have a lot of deep information about that one particular task. But what we're doing right now is working with a task where we have a lot more going on. So we want more behavioral characterization. And so in this case, we're looking at sort of this is our working structural equation model, which is actually probably not really correct anymore, but this was our hypothesis going into it, was looking at how things are related to navigation ability. So one question is, what are the components of navigation ability? Is it, really just like, are these all just working memory tasks? Are these really, executive functioning tasks? What's, how does this all relate? Do these different types of spatial knowledge really relate together? And then how do other factors like personality or risk taking, other things involved in this? And so to test that, we have tested about 250 people across 2 sites, so at UCI and UC Santa Barbara, for about 10 to 12 hours of behavioral testing. So participants did about 40 different tasks altogether, so including 11 virtual reality tasks, but also, which is in our immersive virtual environment. So this is my former lab, which was I was telling people last night, which was condemned. We're now moved into a new building. It's literally condemned. At any rate, but now we're including tests of episodic memory, executive function, personality, small scale spatial ability. So kind of a whole battery of stuff. So we're trying to put this all together in a beautiful giant model. And about half of those folks had basic brain structure and function. So we don't have the same detailed task-based imaging in this situation, but we have more people doing more tasks outside the scanner. And so I could probably give a whole lecture just on what we're finding from that. But we're still sort of a lot of work is in progress with that. But I just want to give you a sense for one aspect of things that we're doing. So I think there's a lot of challenges, both to see what we're finding on the behavioral side, which has a lot of good questions that we can ask and answer. And there's a lot of good questions from the brain imaging, including sort of multimodal imaging. How do we deal with that? So I'm just going to show you one thing that we've been working on, which is using machine learning to predict performance. And so again, how can we, find out who's going to do well at this? So this work was led by Marsha. And so, so what she's been doing is just saying, let's input pairs of T1 images, just their T1 anatomical scan, and just say which brain performed better. And then basically have the machine learning algorithms learn this. So essentially, by doing this pairwise comparison, we're scaling up and amplifying the data. So instead of 8 participants, you might have 28 unique pairs of comparisons. And when they're scaled up to our 108 scans that we have, it becomes about 5,000 data points. And so now we can use this to sort of, just ask these relative questions rather than necessarily the specific answers. Although we can also do this specifically too. So, and so this in this example, we're just using this mental rotation task, which is just are these 2 objects the same or different? Just using mental rotation. And in this case, the true MRT and predicted MRT based on these, again, T1 images is pretty close. It's quite good with a fairly low error. So again, we're doing a pretty good job with just 108 scans to predict performance using just their brain image. And then she's using some backward elimination techniques to figure out which brain regions are contributing from that T1 image. And in this case, from the mental rotation, has found these brain regions are the most informative. And so the most informative ones are the ones where when you remove them from the model, basically the performance goes down the most. So if their performance goes down the most, it means they're contributing the most. And so in this case, it's sort of superior frontal, as well as some other frontal areas. But these are the top sort of 8 regions, and then of all of the sort of 50 ones that we're looking at. So, so it's been pretty promising so far, trying to put this together. And so again, it's something when one could look at different ways. And we're hoping to then look at this, with clinical populations or other things. So we can use these sort of methods to amplify smaller data sets. It could be really useful. Yeah, so I mean, in this case, it's just sort of saying like which ones have been the most useful for the model, and it's just the T1. So we've also done like standard cortical thickness analysis, for example, and some of them overlap and some are not. So some of these ones are ones that have come out in cortical thickness analysis, but it's also finding some different things. So again, because it may not be, because that's just cortical thickness, this is picking up other things too, presumably, and we don't necessarily know what they are picking, what it's picking up on. But there's certainly some connections or standard analysis.
Subcortical or regions.
In this one, I believe it's a subcortical as well. No, so it's a T1. So it's yeah, and then breaking it by ROI is included, I believe, subcorticals as well. So in this case, with the mental rotation, and all the top regions were cortical, but it could come out that they're subcortical, I think. So just to summarize sort of some of those individual differences where we see behavioral relationships between exploration and performance with evenness of exploration and changes with age, structural relationships between hippocampal subfield volumes and white matter structural integrity, as well as the functional dynamics. And we're still ongoing. So like, we're just starting to submit our first like papers, and like there will be just like a million papers coming out of this one. So I think there will be a bunch of stuff coming. So next, I want to turn to something a little bit different and just talk about how the brain tracks distance in spatial versus other types of spatial relationships. And I kind of mentioned this with kind of graphs, but one thing that I'm really interested in is sort of seeing how much of this connection between space, how much of the brain is sort of spatial versus other types of domain processing, and how this connects, right? Are we doing something different when we're talking about spatial processing, or are we tapping into the same kinds of questions and that? So I'm trying to get at this question. So I'll start with this kind of like crazy question, like what does the hippocampus do? I know we're all studying that, but sort of thinking about this for learning and memory. So we know that there's spatial distance coding in different ways that there might be more activity the further you are from something or the nearer you are to something. But we also know it encodes spatial information and distance on social relationships, and then temporal and sequence coding in different ways. And so that's something that led us to wonder about this question of relational processing, which was really championed by Howard Eichenbaum. And so trying to think about how we can sort of test this question experimentally. And so just to mention, some previous efforts we're not the first ones to ask this question by any means. And they found some small areas of parietal cortex using multivariate approach, not much in hippocampus, not much in the univariate approach. And they tended to find more domain-specific processing. So maybe that's just the case. But we just wanted to give it a go because a lot of these previous efforts have also used different stimuli for the different domains. And also they've tended to be mostly egocentric or related to the observer. So you'd ask, how far is this, from you? How far is the ball from you? How far is this friend from you? And not allocentric or independent of the observer. So we wanted to also look at that dimension. And so here we developed this representational distance task, where it's kind of a fun one. We ask people, we talk about famous people throughout history, and ask our participants to sort of give ratings about these famous people in relationship to each other and also into relationship to themselves. We take a picture of our participant and stick them in there. This is Mike, who's doing the study. So you can see right there. And we sort of say, like how far in time, right, are Abraham Lincoln and Robert E. Lee? Well, maybe not very far in time, maybe not that far in space, but maybe far apart, further apart in social distance. And likewise, right? So we have people from sort of around the world and different types of, relationships. We did a lot of piloting to try to get a good spread. And so we just ask them at each trial, again, put a rating scale about how far, you are from a Barack Obama in a spatial relationship. And we leave it sort of vague. We do operationalize social distance as like how you get along. Like, do you have to share the same morals and values? Would you get along together at a cocktail party, perhaps? And there's other ways of doing that. And then again, we can kind of leave it up to the people because in fact, what we care about is more what they say and how they're doing the relationship. And we do see that indeed people on the behavioral level can scale up, right? So there's distance ratings basically from 1 to 7, and everything in between from both the egocentric and allocentric ratings. Their confidence ratings are also quite high. And then when we look at this activity, so now we're looking at activity that's modulated by distance, and both increasing and decreasing. We don't necessarily care. We just care that it's really that the scale, that activity scales with the distance. And we do see areas of this. So we see areas in medial parietal cortex, sort of maybe like fusiform, some early visual cortex, interestingly, as well as some like sensory motor areas. So we do see some regions that are do scale with this domain general. This is across all trials, domain general distance modulated activity. We also look at activity when they're doing the different tasks. So if you're doing a spatial rating versus a social rating versus a temporal rating, we do this sort of like Venn diagram of the three. We do see areas in white, which is when they're doing when they're, the areas of overlap for all three. We see anterior hippocampus is active for all three types of rating, including, and as well as some areas in like thalamus and some parietal areas and early visual cortex, which again makes sense that they're doing this on the task. And then we can also look at egocentric versus allocentric. So comparing ones where it's more allocentric, which would be the sort of orange ones, and more egocentric, which would be the sort of blue ones. We do see some differences. Interestingly, the hippocampus is more egocentric, which we were not necessarily expecting, as well as some of the medial areas are more egocentric, and the other ones are more allocentric. So we are seeing some stuff in these univariate analysis. And then we also have did representational similarity analysis, where we can do basically looking at the coding of domain or reference frame, or the distance coding, or just the face that's on the screen, and just sort of say again, like for these, are we getting these codes in some of these areas? These are ROIs of interest. So whether we can do this sort of similarity analysis, and we do see that in hippocampus and entorhinal cortex, we see basically similarity, but only for distance coding. So not particularly for the domain or the reference frame, but only for the abstract distance. Whereas retrosplenial, supramargus, precuneus, angular gyrus actually also code for domain and reference frame. Nothing is decoding for face in this one, so that's also nice to see. It's not doing everything. So from this, we kind of have a sense that hippocampus is active during the task, regardless of domain, which suggests some abstract relational processing. We see distance coding in posterior medials and visual areas that are, again, across all domains. We see regions that distinguish between face, reference frame, and domain using RSA. And so we're hoping in the future we might learn more about how we learn these relationships, how this happens over time, how we put this together. So these are well-learned relationships, and we always test people to make sure they know everyone. But maybe how do you acquire that? And then maybe seeing whether these representations change during the aging process, whether this could be a signal for something that might happen during aging. Okay, so let's talk about aging and what happens to our navigation abilities as we age. And again, we're looking at the earliest stages of aging here. So we're going to revisit some of these tasks that we've seen before. So you've seen the maze task. I'll bring you two other tasks. Here we are talking about midlife. So earliest time periods, 45 to 55, not that old, saying as someone who's in that area. And we also, of course, during midlife is when women undergo menopause. So we're also interested in pre, peri, and postmenopausal women. And so we're getting both looking at chronological and reproductive aging here. So our first task is that wayfinding task. So I won't show this again, but again, this is the maze task in the scanner. We see behaviorally that our young adults, our young men are doing really well, young women are doing okay, but everyone's kind of doing not very great at midlife. We really see some reductions here. When we start looking at the brain, we're really focused on the white matter tracts in this case. We used this associative memory as a covariate to control for general memory. So what we're looking at was really specific to the navigation. And we find that in this case, performance is correlated with sort of our motor tracks, sensory motor tracks, so cortical spinal tracks and things like that. Versus, if you recall, I mentioned in our younger adults, we really saw these limbic tracks. We also saw sensory motor tracks, but kind of a different set of tracks that were really related to performance here in our midlife group. Now, this is all midlife on the whole. We can also break it down because we had reasonable numbers. So men, when we break it down by men and women, we find that men have negative correlations with the fornix. So that's actually opposite of what we saw in our young adults. So our young adults had this positive relationship with increased microstructural integrity in fornix. Was related to better performance. Here it's actually with worse performance. So that was definitely a puzzling.
But both men and women, again, tend to have sort of these positive relationships with sensory motor tracks on the whole. So, okay, so so some moderate sex differences, but nothing too crazy. We do, again, it's smaller sample sizes, so the treating is sort of highly exploratory, but we do see in our premenopausal women, the fornix is related to better performance, right? So we see a positive relationship. So we see this really weird sex difference between our sort of midlife men and our premenopausal women. Our postmenopausal women don't show anything particular, but they show this sort of same sensory motor track. So again, we're starting to see these changes emerging across midlife, and it really seems to depend, especially on sex, but potentially as well as on menopausal status. In our next one, we're looking at spatial strategy. So we're looking at the dual solution paradigm. In this task, we're really focused on what people do, even if they know where they're going, how they decide to go. So it's a strategy task. So people follow this route 5 times. They learn it really well. And then we ask them to go from one object to the other. In this case, the objects are visible, so they tend to find the object most of the time. But our sort of main question is, if we ask you to go from here to here, do you follow the route that you know, or do you sort of take a shortcut and go through, right? So you might have figured something out more about the maze. And so that's a real question. So in young adults, we've seen that, there's some sex differences, and that if you tell people to take the shortest route, that the sex difference is reduced. So it might matter a little bit there. But we wanted to ask this question again in midlife, and we see again an overall sort of aging effect, as well as a sex difference where generally men are getting there better. But in particular, when this question of shortcuts, only really our young men are taking the shortcuts, and pretty much everyone else is not. So our young women, our midlife men and women are all following the learned route much more so than the young men. Okay, so then what's going on in the brain, in particular in our midlife group? So overall, again, when we combine all the people together, we see mostly motor and sensory motor groups, and this is actually associated with fewer shortcuts. So this is tending to take the learned route, is really sensory motor. We actually don't see any associations with taking more shortcuts. So we thought maybe we would see something in, fornix or limbic structures. We don't see anything. But again, their midlife people are not taking that many shortcuts to begin with. And when we break it down by sex, in men, we find here, if we look at just the men, we do see some relationship with more shortcuts in some of our association fibers, so IF, OF, or ILF, and arcuate fasciculus as well. So some of our sensory motor, of our association fibers are related to more shortcuts in the men, but not necessarily in the women. And we still see this sort of motor areas. And on our women, an overall sample, again, only just relationships with sensory motor and fewer shortcuts. So we're not seeing any real relationships with taking more shortcuts in women. We can also look at some of the hormonal changes. So we do see overall that decreasing estradiol, which is what happens during menopause, is associated with increased reliance on those habitual paths. So people who are postmenopausal are tending to take more of this paths that they know rather than taking more shortcuts. And this is controlling for age as well. So even regardless of your chronological age, it's really related to this estradiol. And then when we can stratify by menopause status, and again, our samples are pretty small, but we do see that in this case now, when we look at our premenopausal women, we see now finally fornix integrity is related to taking more shortcuts, but only in that group. And in our sort of later groups, we don't really see, it's just really again related to sort of sensory motor processing. So again, some places where these things matter. In our third study piece of this, we did a path integration task, which is just being able to update position and information, position and orientation while you move. So we have this virtual desert environment. It's not much to look at because we purposely don't want to have people see much. So it's a virtual desert environment, and we're actually having people walk in this environment. That's the Santa Barbara lab. So what they see is there's 2 eyes here, and they're just walking them in a circle. So we walk our participants in a circular path and just say, click the button when you think you've returned back to the start location. So it's a very simple task to understand, and this is a walking task, but it's actually really, really hard to do, if you can imagine. And so we again looked at just behavioral level. Males tended to overshoot and females tended to undershoot. But But in terms of distance traveled, so we look at our 360, are actually, sorry, yeah, males tended to undershoot and females tended to overshoot. But in terms of like overall like error, they're not that different. A little bit different at this 3 meter loop at the largest loop size. But overall, like men were on one side and women on the other side, but they're about equally far off. They just tended to have a different strategy. So we suggest that this is a strategy difference, not necessarily a performance difference. And then when we look at the brain, we also tend to see not that many differences. Overall, we actually here we have some hippocampal subfields volumes, which is the first time we've been able to look at hippocampal volume. We do see that the volume is related to degrees traveled here. Again, it's a pretty small sample, but we do see that across most of the board, not subiculum, but in the other subfields, that the volume is related to this like strategy. We also see, and again, correcting for all those intracranial volume and stuff. We see here that the fornix is related to degrees traveled in midlife overall. We're not going to stratify by, so I think we're going to do a little sex stratifying, but and then we see, but in terms of accuracy, so in terms of, again, this distance traveled, whether you tend to overshoot or undershoot is where we're seeing the association fibers. But just in terms of like how far you get to the target is really, again, our sensory motor tracks. But our young adults actually do a very similar thing in terms of the angular error. We see almost the same tracks in our younger adults as we see in our midlife adults. We don't see anything actually with distance traveled for our younger adults. So, but overall, we don't see very much age difference or sex differences. And again, we don't have as much sample to like separate out, and we really don't have sample to separate by menopause in this case. But we're seeing places where there are not as many age differences happening. So our next steps, we have this more complete battery of aging, including immersive VR. So we just got funded to start this project. So we will be launching this. We're launching this right now. So we have now we've sort of taken our lessons from our younger adults and to do a more comprehensive battery. Everything's now an immersive, and some of those were in the previous one were on desktop, which helps with reduced motion sickness, especially for our midlife people, because we were seeing a lot of dropout from motion sickness, improves proprioception and vestibular cues. So we're really excited about that. We also have a hormone assay and AD risk factors. So again, getting a lot more information from folks, and with an imaging session of basic structure and function. And oh geez. Okay, I'm almost done. I'm out of town. But I just want to mention another little aging thing that we've looked at. This is work with Kevin Byers' group on the earliest changes in Alzheimer's disease mice models. And I'm not going to talk about like 95% of that work because that's all his lab, but I'll just tell you the bit that we did, which was looking at the ADNI database, comparing white matter tracks between hippocampus and retrosplenial or entorhinal cortex. So, because again, people are always looking at entorhinal cortex as the earliest area, and some of his work has been suggesting that retrosplenial cortex might actually have an earlier signal in terms of hyperexcitability at an earlier onset. And so we've been looking at the ADNI database, and we found, indeed, when we compare the tracks, white matter tracks going in retrosplenial area versus entorhinal, that especially in females, we see these differences such that people with MCI start to see significantly different changes in their white matter for females, but not for males at all. But that entorhinal is happening at a later, so only the full on Alzheimer's group shows differences and not the MCI group. So we start to see increases, at an earlier time here. It doesn't seem to matter by amyloid status. But again, we see an age relationship in retrosplenial, not in entorhinal. So just sort of starting to look at this area again as a potential early target. And retrosplenial is like my little pet region. And so I'm always interested in finding more about that. And we're doing a lot of work starting about that. So anyway, we're continuing to do some of this stuff. We're looking at exploration behavior in Alzheimer's disease. We're looking at some sleep studies, hopefully coming up. Again, hoping to do some sort of longitudinal work. What is it about navigation, right? So if you're bad at navigating when you're 25, some of you, like, are you going to get Alzheimer's? Maybe, maybe not. Or is it a particular aspect? Is it a change? That's something we want to find out. So which aspects are most relevant for Alzheimer's risk? And I'm just going to roll through this. And I don't know, I don't really have time to talk about this, but I did this kind of crazy thing when I was pregnant with my kid. I scanned myself a bunch of times before, during, and after pregnancy. So it's kind of a different aspect. We just want to mention this because we see this huge changes in sex hormones. So we're really interested in, what's going on here with us, all this menopause work. So what's happening during pregnancy? So it was a really great time to do that. And so we find that there's these huge decreases in gray matter volume throughout the brain on the order of like 8% on average, with a small rebound postpartum. So like here's, so you're like average, these are ventricles, but we see some changes overall with gray matter volume, a little bit of a rebound postpartum, but not fully. But interestingly, we see some areas that sort of improve, if you want to call it that. So white matter integrity showed increases that sort of peaked in the second and third trimester, but then returned to baseline. So if you do sort of the pre and post, you would have missed that. And so this was able to find some of that work. So, okay, so just to summarize all this sort of stuff, we talked about aging, individual differences, some distance tracking, again, aging and menopause, and then life transition. So I will just wrap up and say thank you so much for your attention. Thanks to all my collaborators, my students, funding sources, and everything else. So I really appreciate your time. Take a few questions. Mm-hmm.
Given that the age gap between them is not very great.
Yeah.
But I'm wondering how are you separating out the effects of age
Mm-hmm.
from
From which effects?
cohort
Cohort effects. Yeah.
effects, particularly
Mm-hmm.
to be pretty strong. And I'm wondering how you know whether these cross-sectional differences that you're seeing
Yeah.
really be attributed to age as opposed to
Just like older midlife adults aren't playing as much video games and other things, or other kinds of experience, education. Yep.
experience video games,
Mm-hmm.
lifestyle effects
Yep.
change over. So
Yeah. So I think,
that the-
yep. I think,
how do you separate that?
yeah. So that's one thing we were not able to really do as much with this current setup, is because there are these interesting life effects. One thing that's really important, I think, is that, our midlife and older adults will have like didn't grow up using GPS, and our younger adults like now have like all used GPS growing up. And so I think that's a big one. And so one question, we're hoping, there's lots of things we would like to do. So some of it's going to be longitudinal, right? So some things are going to be, again, when we look at the midlife, we're hoping to oversample our like early aging so that we can then bring people back in so that we can do some of these aging things. And so that's, again, we're trying to get some of the more longitudinal studies in our older adults. I would love to start people at like 25 and just bring them back every couple of years for the rest of their lives. Well, you know how that is. So some of it's going to be doing some more longitudinal work. Some of it's going to be, some of it could be experimental, right? So things like changing, some of these, like training people on video games, training people on taking away their GPS, doing, stuff like that. But then other things are just finding out more about that, including that as covariates in different ways, right? So, and that's probably the most like practical and simple way to do it, is just to find out more. So, and unfortunately, we have this video game questionnaire, which normally goes in almost all of our studies, and somehow, for some reason, it didn't get into that midlife one. But we are now we make sure that goes into everything. And GPS use is another one that we ask. We also ask about life experiences with like did you read maps a lot when you were a kid? Do you go hiking? Stuff like that, like just like your like experiential factors, which interestingly, at least in our young adults, did not come, only really video games came out as the only thing. Yeah, so as related, even though like they actually do a lot of things like we call it like in-person shopping and like learning in new places, but they might do it in a different way that doesn't actually tap into navigation as much. Yeah. 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 We will be in our upcoming study. So we will be doing lifespan. So we'll be at 18 to 80. We just, as it turns out, as the way we collected that one, our sort of midlife study, it was just kind of the groups that we had going, and we just try to put it together, but we will be.
Us with that. So to what extent, and I think first of all, it is a great short study. Sense. But to what extent does the virtual reality environment reflect actual information? Do you have any data like that vary across individuals or cohorts?
Yeah. Yeah, so I think again that's a really important question. So that's one reason we certainly want to make it the ambulatory as much as possible, right? So to at least eliminate the controller effects. But yes, there is something, it's a little different, and just some of the environments are different, right? So we tend to use like these more maze-like environments. And that might be a little different from navigating a city. And so we sort of know that, and sort of that's our paradigm. But we've, been looking at that in other ways, and we're trying to test that in mostly in younger adults, but I think it's important for older adults. And we've also occasionally do work in real world. So we try to balance the control of a laboratory setting with the ecological validity. And so this is our best measure, but I definitely agree that that we don't totally know, like, does this mean the same thing as, navigating around town? So, I think that's some of these questions, right? So I think, some folks have been working on this like phone game, right? So we're using a phone app, and that's something that I've found very challenging. And so that's one reason we were including that one in this study, because that's even more removed, I think, from other things that we're doing. And again, I feel like I'm a good navigator, and I'm just like, I cannot do this phone app because it's just like too wild and different from things I do. The See Hero Quest one, yeah. Yeah, so it's a video game, right? So if you just aren't good at it, and I also got like really motion sick and other stuff, right? So like, but maybe, there's different ways around it. So we're trying to like optimize as best we can the naturalisticness without and have it be real, as close to real world while still having the control. But yeah, it's still, but but exactly, but the environment is a little different, yeah, to control for familiarity and all this other stuff too. So, so we've also, we're trying to include things that might be, different styles and different types of environments to try to get at a broader sense for that. But yeah, it's not going to get at everything, and that's definitely a limitation.
Two questions. I'll get to try and be quick about it, but talking beginning about different cognitive strategies. I'm wondering if you looked for sort of singular types of brain representations of like optimal accuracy, but have you considered looking for biotypes that might reflect cognitive strategy?
Yeah, I think that's a really important thing. And that's actually some ways that the bigger picture inspiration for some of these individual differences work. So we actually have a side of this, which is actually robotics. So so with the idea of like, okay, if navigation is so important and critical to our lives, shouldn't we all be doing really well at this, right? So, but perhaps there's some inherent advantage to the variability on a population level, right? So perhaps having multiple strategies that some people are sort of good at doing this way and doing that way, that might be useful, maybe not on an individual level, but on a population level, that sort of keeps us more balanced. And so that's one question, that's sort of the big question driving some of this work, is to see like, if we then put in sort of different strategies, does this actually help? And so in the robotics side, we've actually done that with our collaborators, and they've sort of modeled having different kinds of strategies, and that does seem to like have increased coverage and decreased time to find things and like that. So having different strategies is helpful. It wasn't maybe the most ideal implementation, but I think that's a question. So I think, again, thinking about, yes, performance versus ability versus strategy are all kinds of things that are involved in this, and I think that's a really critical thing, that it's like not necessarily what you could do under the best circumstances, but like, what do you do today? Yeah, so I think that's really important.
Second question, separated. Estrogen has both a key organizational effect. It seems like you're considering this to be like a lasting organizational impact. But I was wondering if you looked in your healthy younger adults that menstrual cycle days to see if you see a similar change between molecular.
Yeah, so yes, I mean that's super important. So cycle studies are something that we haven't done in the lab, but some we're collaborated work. So like Mary Hegarty's student has done that, and the student is now coming as a postdoc in my lab. So we probably will be doing that. And she has found some differences between cycle phases. And so that's a big question. Yeah, sort of, yeah, there's like those organizational effects and the fluctuating hormones. And so I think that's important when we talk about these sex differences, right? And everything. It's like there's so many ways that this could come out, right? So it's like organizational effects, fluctuating hormones, socialization, other things, right? There's just like chromosomal things that can, that might affect organizational changes, right? So there's all kinds of stuff that can be going on, and we don't necessarily know where any of this is happening. And so I think those are all important to consider.